This is the Science Podcast for March 12, 2026. I'm Sarah Crespi. First up, freelance journalist Evan Howell takes a trip to Cape Blossom, Alaska, where eroding cliffs have revealed an ancient glacier that may offer a glimpse into a pivotal chapter of Earth's climate history. Next on the show, machine learning, machine vision, and data compression have come so far that an entire fish's life can be captured and analyzed. Researcher Claire Bedbrook talks about using this approach to predict lifespan in a short-lived fish. Finally, Matthias Loreto is here to talk about tracking the relationships between wolves and ravens in Yellowstone Park. His team was surprised to find that instead of following wolves around and eating their leftovers, these scavenging birds use a different strategy to find unpredictable food sources. I saw you're a geologist turned journalist. So was this a story that you were super excited about because of your geology background?
When I worked in geology, I worked in really, really old rocks, hundreds of millions of years old rock. But I always had this fascination with the Arctic. I think that was what I was thinking I wanted to do when I first got into geology. I loved big mountain cirques and alpine terrain. And so glaciology kind of fit in with that. But for whatever reason, my interests academically went in a different direction. And so I studied very old rivers and things like that. When I started writing, I just kind of latched on to this world, the cryospheric world, as they call it. Got invited up to Alaska, which I'd never been to, so couldn't say no to that.
Which is very cool. But this is just baby stuff for you. 350,000 years is nothing on the scale of rocks, right?
It's true. Yeah. In terms of deep time, this is just scratching the surface. When glaciologists talk, they talk about deep time being hundreds of thousands of years, but it's all relative. It depends on what you're trying to understand.
If you want to get freaked out by scales, just go talk to a geologist.
Yeah. I can go on about that for a long time. Yeah.
This is Evan Howell. He is a freelance science journalist and former geologist. He actually went up to Cape Blossom, Alaska. It's really far north, and it's not easy to get to in order to see this potentially very, very ancient glacier. So I asked him, what was it like going up to that part of Alaska?
This is not somewhere most people go on a tourist trip. A lot of people, when they go to Alaska, go to the fjords and the glaciers down in the south near Anchorage and near Juneau, which I did too. But going up here is a much more different experience. It's a very remote part of the state, and it's just within the Arctic Circle. So I got the full experience of like the very long days, the Inuit culture and everything like that that's up there. I mean, this town, the village of Kotzebue, I believe, is home to about 3,000 people, hundreds of miles from the nearest road. Everything has to come in by boat, plane. It's just a very remote place. Everyone's driving around on little ATVs.
And you're there to basically talk to some glaciologists, some scientists about ice that's starting to be revealed by the erosion of the coastline up there. So that's a result of climate. But what it's showing is that there's this glacier that might have lasted hundreds of thousands of years and can tell us something about these kind of climate highs that we might be coming up to. So how did people first figure out that there might be some really, really old ice in Alaska?
At least by the 1980s, one of the main characters in this story, Julie Brigham-Gretty, she's actually admitted to seeing ice in the area at the time, kind of poking out of these bluffs, but didn't really know much about what to do with it. The analytical techniques on ice weren't nearly what they are today. She's like, oh, that's interesting, but didn't think much of it. And then Ben Jones, a geographer at the University of Alaska Fairbanks, just a few years ago, was kind of flying around in a helicopter, noticed some unusual soil textures, and was like, hey, that looks like maybe a whole lot of ice down there. And so a year later, they came in on foot. Because again, this is a very remote area. You can't just drive up. So it's a big deal to come out to these areas. You need helicopters, you need boats, something like that. So he finally got around to checking it out, and they're like, whoa, this is a big chunk of ice. This doesn't look like the more common permafrost features we see up here.
How big do they think this glacier is that's buried in the cliffs here?
That's pretty much unknown at this point. If you fly over, there's a video that will be on the website that I took flying in on the helicopter, and you can see that Cape Blossom has this like kind of anomalous sort of hill shape, tilted hill. Otherwise, there's this very flat tablelands. The whole region is like flat as a board until you get to the mountains. But that kind of hill suggests that maybe it's being propped by this ice. So maybe it's the extent of this hill. But otherwise, we don't really know. It's actually kind of hard to image through all the permafrost. There is a lot of ice in the ground, and that's worth mentioning. It's the Arctic. There's a lot of permafrost. There's these big wedge ice features. But they're like, this doesn't look like that. But they're not glaciologists, so they wanted to bring other people who are. And so that's where they kind of put together this field trip. And I got invited as a writer to kind of be a fly on the wall with these sort of things.
This is a glacier buried underground, and you're just seeing the toe of it start to be revealed by the erosion of the coast. So, kind of now it's like, well, what is it? How old is it? When did it get here? What can it tell us about the past? But that's really not easy to do. Let's talk about why. And I think some of that plays into this difference between ice in the northern hemisphere and ice in the southern hemisphere. Can you talk a little about the differences there?
Northern hemisphere versus southern hemisphere, almost everything we know, or most of what we know from ice, which is a very precious climate archive because it traps like fossilized air. It's like a mosquito in amber. It is like the air that existed at whatever time, whether it was 20,000 years ago or 200,000 years ago, that it captures in that ice. Now, Antarctica is unique, especially in the interior, in the highlands, because it is just damn cold. It doesn't get very warm. So it's just been piling up layer cake year after year for hundreds of thousands of years. Every new winter brings more snow, which compacts into ice. Now, in places like the northern hemisphere, I mean, even Greenland is much warmer than Antarctica. So its continuous ice record only goes back to the last kind of warm spell, which is about 123,000 years ago, I believe. Whereas in Antarctica, they have a continuous record of 800,000 years and ice up to, I believe, millions of years old, maybe 6 million years old now.
Antarctica, you're seeing 800,000 years, whereas up north, it's like one glacial cycle.
Yeah, there's a lot of great science coming out, especially in the north, showing that Greenland, other areas, when it got warm, these ice sheets really melted out. Maybe not entirely, but at least a lot. And even in Antarctica, a lot melted out, but there's always been a big core that remained intact. And Greenland probably did too, but a lot of it melted. And so we've had these interglacial and glacial cycles dating back the last 2.5 million years. A lot of people don't realize that. I mean, we've had warm spells naturally prior to this that were very much like today. That's why folks were interested in this Alaskan ice, because that gets back to a time, it almost sounds like a James Bond character, MIS 11. It's like, so these marine isotope stages, I'm not going to go into the details, I bore everyone, but that comes from marine records of all these isotopes in little plankton and foraminifera. So they understand that there are these warm and cold cycles through time over millions of years.
You can see this from the bottom of the ocean where layers are building up. So it's not about ice anymore. It's about the animals and the chemicals that were kind of trapped over time. And we can kind of say, this is what it tells us about climate, but it's not going to be the way ice is going to be informative.
It's a proxy for ice volume because when ice sheets grow, they lock up one form of oxygen isotope at the expense of another. And so you can see that ratio changing in the marine sediments, which is pretty wild. You're taking cores out in the middle of the ocean, which is telling you something about ice volumes in the poles from, thousands of miles away.
So it's a proxy, but we could get better. We could be better.
It's kind of an average look of ice volume. But what they really want is like, what was ice doing in various places at different times? Because I've put in the article that it's kind of like a poorly rehearsed band. One member of the band's playing one chord and the other playing another. So these ice sheets don't grow in sync throughout the world.
There's local climate.
Exactly. What we learn in Antarctica only can tell us so much about Antarctica for the most part. There are a few things, a few gases that are well mixed throughout the whole globe, but not everything.
I think it's important that you point out in your story that 90% of the population is in the northern hemisphere.
Correct.
We do want to know what's going to happen with climate here, and a peek back at former heating and warming cycles from northern ice would be very useful. But now that we've found this glacier that I'm going to say is potentially very, very old in Alaska, we had a little bit of trouble with actually finding out how old it is, kind of looking for distinct layers. Can you talk a little bit about some of the troubles that scientists are facing, basically sussing out this ice?
Right now, we don't have a specific date of the ice itself. The ice's age is based on a relative age of the sediments that surround it. These glaciers were kind of like, if you go to Juneau today, you can see these glaciers growing into, they call them tidewater glaciers, growing out into these beautiful fjords. They come down out of the mountains and into the sea. And so that's what they believe this one was about 350,000 years ago. And that's based on some age that come from the sediment. So it's a proxy. I mean, this stuff is almost 40 years old, these dates. So no one's really done that detailed work in a long time up here. And so when they found this ice, they're like, well, because it's in these sediments that are roughly dated, we think this ice must be roughly this age.
And we were saying 350,000, which is twice as old as anything else that's been thawed that far north and, getting us through a couple glaciation cycles.
And that ice is sitting kind of within and above some sediments that are associated with a very long and very warm period, much like ours today. So that's kind of interesting. MIS 11, like I was saying earlier, is one of the longest and warmest interglacial periods that we know of during the ice ages. So people are very interested in what's going on then. Like when we get that warm, how much ice melts or how much the sea levels rise. We obviously want to know that because we're heading towards temperatures that are very much like those of MIS 11.
It's not a layer cake like the beautiful Antarctic striations that people are excited to look at. So, what do you see when you look at this glacier?
When you take an ice sample in Antarctica, you drill a very long continuous core. Now, this stuff's just barely sticking out of the muck. So, Philip Wilson, he works with Ben Jones at the University of Alaska Fairbanks. I mean, we have photos of him just cutting this stuff with a chainsaw. He's just cutting out a block. So, this isn't a continuous core. They just wanted some first order information. It's going to be almost impossible at this point to kind of have some layer cake kind of continuous core. They just don't have that. But because this ice is in the north, because it was warmer than Antarctica, this ice appears to have melted during the summer. 350,000 years ago, it was melting. So a lot of this gas was freed and then refrozen. So it's not that, at least it doesn't seem to be this native, pristine, fossilized gas. It's almost 2/3 of the content is methane. That's very high. We would not expect that much methane in the atmosphere naturally. So it seems that it was probably as this glacier is grinding along, it's picking up stuff from the ground, rocks, it's entraining microbes. It's warm enough that those are kind of producing their own biologic activity. So you actually get methane forming in the air within the ice. It's kind of a messy, contaminated snapshot, is kind of where things are at this point on the preliminary data.
And what about dating it? So the gases will tell you something about the date.
They can use Krypton 81 or Argon 40 are two methods you can use to date the actual gas within the ice. And those results should hopefully come in within a year. There's some analytical backlogs. But then the water isotopes, those can be used to kind of establish a temperature at which the ice was forming, which is very useful for climate models. We'd really like to know what the temperature was when ice was present or not present. But again, that's because it's this messy kind of melted and refrozen ice, that's going to be rather complicated, at least with this very small sample they have so far. Now, maybe if they go and find another chunk with a lot of gas, maybe that opens up different possibilities, but it's all kind of early days right now.
Is there a sense of urgency that, this coastline is eroding? One of the researchers you talked to was like, I was here before, the coastline is a lot different. Are they worried that this opportunity to look in the past is being erased?
That's pretty much the essence of what everyone says when you ask someone about this, like how rare and precious and how soon this could be gone. And having been there, I can attest to it. I mean, these cliffs on a warm August day were literally melting before your eyes. This stuff's just falling. We had to be really careful to not get hit with massive chunks. It's a remarkable amount of erosion each year. I don't think we can really comprehend how quickly, because this ground has been held together with permafrost. Warming in the poles is amplified like 4x compared to what we experience in the lower 48. All that permafrost, that glue kind of holding this very loose, unconsolidated sediment is just rapidly melting. It's just kind of turning to muck. And that's what it felt like. And that ice is obviously being exposed, sitting there in the open sun, and it's going to melt. It's going to either be eroded by the coastal waves or melted by the sun.
This is kind of like, I guess, a curtain raiser, they call it sometimes, when they talk about a study that's being done as opposed to a study that's given you results that you can then analyze and kind of push out into the future. So is this just the beginning?
This is the beginning. It's funny you bring that up because I sat on this for several months thinking I needed to wait on results to pitch the story. And then I was just talking to another writer and she's like, no, this is like a really interesting, like the race to understand it before it's gone kind of story. And so I was like, well, maybe, yeah, let's try this. Actually, I think there is kind of this intrigue on what we can learn. And it's just this messy analytical thing that a lot of ice scientists pointed out are like, we're going to be seeing more of this because there is warming. We're going to be exposing ice maybe up in mountain glaciers that we don't understand. We've always dealt with this. If people call it like well-behaved or traditional Antarctic ice, it's relatively easy to run our analytical methods on and understand, but we're going to be getting these messier, complicated archives that we need to kind of retool to understand and glean information from. I think the other thing worth mentioning is that, so if they can say that ice was not only here, but exactly when and have a date with it, that's really powerful information that modelers like Roger Creel, who's at the Texas A&M University, can put into sea level models and try and see, like, it's really more powerful to say ice was not only here, but when it was here. Because that's really useful in these kind of sea level models and projecting into the future, of course.
All right, Evan, thanks so much for talking.
All right. Thank you, Sarah. Appreciate it.
Evan Howell is a freelance science journalist based in Colorado. You can find a link to the story we discussed. I'm sure there's diagrams, maps, and a video that you can also look at science.org/podcast.
So good, so good, so good. Everything you want for summer is at Nordstrom Rack stores now, and up to 60% off. Stock up and save on the brands you love like Vince, Sam Edelman, Frame, and Free People. Join the Nordy Club to unlock exclusive discounts, shop new arrivals first, and more. Plus, buy online and pick up at your favorite Rack store for free. Great brands, great prices. That's why you Rack.
Ready to soundtrack your summer? With Red Bull Summer All Day Play, you choose a playlist that fits your summer vibe the best. Are you a festival fanatic, a deep end DJ, a road dog, or a trail mixer? Just add a song to your chosen playlist and put your summer on track. Red Bull Summer All Day Play. Red Bull gives you wings. Visit redbull.com/brightsummer ahead to learn more. See you this summer.
In other stories on the site this week, we have a lot to talk about from news. First, I want to call out this story by Selena Zhao. She wrote an in-depth story about how to tell if an AI would make a good scientist. What are the benchmarks? What are the tests? How do you show that they can do all of the tasks needed to produce good science? And of course, the news site has a lot of animal stories that I want to touch on. How bees avoid drowning, pre-Inca parrot trade in the Andes, why rodents gnaw, maybe they enjoy it, and finally, ants navigate using a newly discovered type of moon compass. So that's all on the news site this week. In Science Robotics, there's a paper by Alexandra Mihalko and colleagues on using an upper arm exoskeleton to better sync up violinists playing a duet. The team showed that adding bidirectional haptic feedback, so both are getting a little bit of feedback from their partner through the exoskeleton, improved coordination between musicians. Finally, in this week's Working Life column, this comes out through our Science Careers vertical, and it's essays written first person by people in science, scholars, academics, industry. In this one, a professor debates the pros and cons of hiring a graduate student or maybe employing an AI in his lab. Stay tuned for 2 more segments in this show. First, one on what we can learn about lifespan from filming the entire lifespan of a fish. This week in science, Clara Bedrock and colleagues recorded the entire lives of African turquoise killifish, tracking behavioral differences between short and long-lived individuals, and found some interesting patterns in their lives and aging. Claire is here to talk about those results. Hi, welcome to the Science Podcast.
Thank you so much for having me. I'm really excited to be here.
Reading this paper, it takes you a minute. You're like, oh, they filmed fish in a tank for their entire lives. It's an accomplishment for a number of reasons. And there are some obvious challenges to studying an entire vertebrate animal's life, learning about aging, learning about lifespan. The time it takes to do that is the obvious challenge. But what other kind of less obvious challenges do you face studying something like this?
Data size, dealing with, we're recording at 20 frames per second. We've been having recordings go for years now. So it is a challenge to work with that magnitude data and interpret it and get meaningful information out of it. The calendar time, as you said. And one big thing I think is that aging, part of why it's so interesting is it's highly variable and complex.
And the fish you studied are very short-lived for vertebrates, like probably the most short-lived thing that people keep in captivity. But it turns out when they, live on average or they spend about 250 days in your study, that's an enormous set of data, 20 frames per second over that time. What kind of technologies did you use to help wrangle the size, the volume, but also help you get insights into this big data set?
We're using deep learning based methods to go from a video of a fish to actually, we just track 6 different points along the animal's body. And that really gives us the ability to track it throughout its entire life. But that in and of itself is a huge compression going from, a large pixel by pixel video to just 6 different key points to describing the animal as it moves within its tank throughout its life.
And then you used more learning models to kind of turn these into behaviors that could then be used to say something about what happens to these fish over the course of their life. So you're using observation of behavior, and this amazingly was able to predict their lifespan. So once you had enough data on enough fish, you could then put individuals into different buckets, like a long life bucket and a short life bucket, depending on their behaviors early in their life. So you could say, once they've reached maturity, we see these different behaviors. This one's going to live long. This one's going to live short. So what were some of the behaviors that were keyed into that linked short life to behavior, long life to behavior?
So this is at relatively young age, so before middle age. These are behaviors that animals exhibit that are different depending on if they're going to be destined for a long versus short lifespan. There are many different behavioral changes, but some that I think are probably most relatable are just actually movement velocity, and especially peak movement velocity. Animals destined for a long lifespan show higher levels of movement velocity, especially sort of sprint speed. Another big difference is both how much the animals sleep at this age and the timing of that sleep. So animals destined for a long lifespan at middle age are showing quite a lot of sleep, but it's really more restricted to what is their night period or the dark period of their 24-hour day.
I would hate to put this in human terms because slow moving and lots of naps is kind of a lot of people's preferred lifestyle.
Yeah.
That's super interesting for fish, for these fish.
Yeah.
I want to call out the fact that you're observing behavior as an indicator here, but that means you don't have to take samples from your fish. You don't have to sacrifice them. You can just link this, and then it can then become, once you kind of connect behavior to this outcome of lifespan, you can then use it as a marker for interventions. Like if you wanted to change something about the fish, you don't then have to wait till it's dead to say something about its influence on lifespan. So there's a very cool kind of system you set up for studying aging, for studying lifespan. And one of the places you kind of wanted to look was along these time points and say, well, what's different about the inside of the fish if they have these different behaviors and they have these different lifespans? And then you did this as transcriptomics. What did you see was different between the long life and the short life fish?
At a relatively early age, when we looked at the transcriptomes of the animals that, based on their behavior, were destined for a short lifespan versus long lifespan, some of the big differences we see are metabolic differences. We see differences in ribosome biogenesis. So we are thinking maybe protein synthesis, things like that are altered. These processes were elevated in the animals that were destined for a short lifespan. Animals destined for a long lifespan look like they have lower levels of protein synthesis or ribosome biogenesis and higher levels of things like autophagy. So turning over, cleaning up, those are some of the differences that we've seen. Something that kind of puzzled us and wouldn't have necessarily been my initial hypothesis going in is a lot of the time with age, when we look transcriptomically, we see big differences in sort of inflammation. That's actually not something that we see to be really different at this early time point when we're contrasting the animals destined for a short lifespan versus long lifespan. So suggesting maybe that's not, in this case, the driver of the difference between these two populations.
Another important finding that we should talk about is you saw these kind of stages of aging. So, with humans, we're like, okay, we got embryo, then we got the baby, then we got the kid, and then we have like puberty, and then you got middle age, and then everything after that is kind of a continuous decline, I would say. But
it's a little dark.
I think there are some insights into this in people we can get to later. But for the fish, what you saw was kind of a structured, like an architecture of aging. There are these abrupt changes that they don't go backwards from. Can you talk about that part a little bit?
Yeah, so we were really struck, and this, again, probably wouldn't have been my hypothesis going in. I would have thought that it would be a really gradual process. But when we just look at kind of whole lifespan behavior for an individual animal, we saw these sort of stark transitions where animals would go for long periods of time where each day to the next were very similar to one another behaviorally. And then there would be a pretty abrupt transition and they would shift to kind of a distinct behavioral life stage is what we're calling them, where they're exhibiting a different combination of behaviors throughout the day. I think this is something that we see really across the population. And we think that these stages that we observe are stereotyped. So kind of similar combinations of behaviors animals are doing across animals. We see similar life stages, which are kind of made up of similar combinations of behaviors. But when an animal transitions from one stage to another is quite different across the population.
if it's like proportional, the proportions are different. Like it's a different slice of their timeline that's spent in these different stages.
Exactly.
Do the long-lived fish spend more time kind of in the end zone, in that last stage, kind of maybe perhaps the least pleasant part of their lives, I'm going to guess?
Yeah, so there is variability across the population. So you will see exceptions that are kind of the really late in life healthy animals. But a large fraction of the animals that live for a really long time spend a long time in these, what are probably less desirable stages to be in.
I just say that because one of the key behaviors is like floating around on your back, like not doing something.
drifting.
Obviously doesn't look good for even a non-fish person. Yeah.
We're excited about it because what we would love to be able to do is not just, extend lifespan, but when these positive periods in an animal's life.
Yeah. Figuring out when they are and when the transitions happen and what's driving them can help with that. Absolutely. You did do some interventions here, like calorie restriction is one most people probably were familiar, like is associated with changing lifespan. And when you did that in the fish, did you see the behaviors change and their lifespan?
We did dietary restriction from adolescence only up until about midlife. So we didn't do the full endpoint to get the actual death of these animals. But during the period that we did record them on dietary restriction, we saw large changes in behavior. The animals looked more youthful based on their behavior. One of the kind of most striking differences, I think, is their sleep behavior, for example, again, being much more restricted to nighttime or the dark period for these animals compared to throughout the day.
So they look behaviorally younger, longer in life.
We've really focused on earlier in life to just see, do we see behavioral differences and do they look more youthful? Do they look older? And it looks like they look more youthful. But I think a really exciting future direction would be to do this intervention, see how it changes those late in life stages. Does it shift them? And then also intervening at different times during life. Can we change the aging trajectory in animals on later points in life, or when is it too far?
So how do these findings here in this very specific short-lived fish line up with what we know about other animals, other vertebrates, people, mice? Are some of the things that you were able to see in this work already correlated with lifespan?
The stage model that we observe, I think there's really exciting data in humans that is suggesting that this could also be the case in humans, where it's not just a constant, gradual, linear decline, that there are these sort of abrupt shifts. And that's been seen in a couple of different studies now. And really, more broadly, of course, across many different species, there are shifts in behavior across age. I think what we're really seeing here and using the fish, that behaviors are really non-invasive but good readout of where an animal is in its aging process. I would be so excited to see that kind of translated to human data. Many of us are wearing watches, things like that, that track our movements. Could that help us inform where we are in our aging process?
Because it's not just about the years, like there's variability there, but maybe there are these plateaus that you could be visiting for shorter or longer times, depending on your background, your genetic background or whatever.
Yes.
And what about doing this kind of study, this holistic video study with an animal that does live a little bit longer? Do you feel like there's a limit? Like rats are like 4 or 5 years.
There is work that is going in that direction that I think is really exciting. I think mice would be maybe the
more attainable,
slightly more doable, depending on how long your grad students and postdocs want to stay in a lab for.
Right.
I think it's definitely doable. What we can do with the fish is maybe we can do it with more animals and start doing more perturbations, things that might be more challenging to do at scale in other mammalian systems.
A lot of stressors, I think, impact lifespan, and it'd be really interesting to see how that mechanism might work.
Starting intervening at different points in life, either with a stressor or some positive change. So
you could be positive.
Yeah, I don't think it is, but I'm saying other.
You could go a positive direction with that. You don't have to stress the fish out. You could give them treats and see if that does things for them.
Yeah.
One thing kind of caught my attention was this idea that as you age, you hit a plateau, you hang out on that plateau, Then there's this abrupt change. We'll say you're going up on the next plateau, and how it could be kind of like this confluence of changes in the body, energy levels, thinking about that, like it's a strategy the animal is taking on to deal with whatever. It really made me think why it's not reversible.
I should be clear, like we don't know what's driving these shifts. So it's hard to say something very strong about it. But my sort of feeling about it is it's the animal kind of resetting their homeostasis based on maybe gradual changes at a cellular level, just get up to a point where they need to kind of shift gears or reset. We don't know what's driving that. We also don't know in our setup, it looked kind of irreversible. They really shifted from one to the next, but maybe with interventions, it isn't. Maybe you can go backwards, but these are things we are excited to explore now.
Absolutely. All right, Claire, thanks so much for talking with me today.
Thank you so much. It was really fun.
Claire Bedbrook is a postdoctoral fellow at Stanford University currently, but over the summer, she'll be starting her own lab as an assistant professor at the Louis Sigler Institute for Integrative Genomics at Princeton University. You can find a link to the paper we discussed at science.org/podcast. Don't touch that dial. Up next, we hear about how ravens find wolf kill sites in Yellowstone Park. Scavengers like crows, ravens, vultures were long thought to follow predators and then scavenge their leftovers. But this week in science, Matthias Loreto and colleagues used long-term tracking data to show ravens actually remember and return to wolf kill sites instead of following wolves around. Hi, Matthias. Welcome to the podcast.
Hi, Sarah. Thanks for inviting me.
So this idea that scavengers like ravens would follow a predator around, is that what you were expecting to find when you started this work, that you would see, there goes a wolf, there goes a raven. When the wolf kills, raven is on the spot?
When we started the study, we really expected that ravens would follow wolves because this has been reported since decades from wolf researchers. And this is exactly what they could observe in the field, at least over short distances. So imagine wolf researchers were out in the field watching the wolf behavior, watching wolves kill something or moving wolves. And there were often at least some ravens around. And once a kill happened, within the first hour, lots of ravens arrived. And this made the quite plausible assumption that ravens must stay close to these wolves.
Yellowstone National Park, why choose that place to study wolves and ravens?
I chose this place because there's a long-term tracking study in Yellowstone. So this is probably the place where we have most knowledge over 30 years of tracking wolves. At the same time, they have since many years a cougar project ongoing. So this was just a place where we had already lots of information, and it was just the perfect setting to add ravens into this system.
So you did look at tracking data for wolves and cougars and ravens. Why was it important to include cougars?
There are many reports about potentially a special relationship between wolves and ravens. There was nothing about between ravens and cougars. So they have in this study system, in Yellowstone National Park, wolves and cougars have pretty much the same prey. So we were wondering whether ravens will also maybe follow cougars, and just nobody has observed it. So we just wanted to compare these two large carnivores.
Cougars are a lot more elusive. You're not likely to see them eating their prey or taking their prey, but maybe ravens are better at following those things than people are.
Exactly. So we thought maybe ravens really can follow the cougars, can find their kills much better than we thought. But it's just cougars have a very secretively lifestyle. So it's really hard to observe. And so maybe we missed something.
So when you looked at this data showing the position of all these different animals over time in Yellowstone, did you ever see a raven following a wolf?
We saw it, but just once in 2.5 years. And this was really surprising for us. We expected this to happen much more often. We found this one case, which means it's possible. And I'm sure it happens more often because we didn't track all the ravens at the time. But it's not what they usually do. So usually they show a different behavior, a different strategy. And we never found it with cougars.
You never saw one following a cougar. And the preferred strategy is just to go back to the kill site that the wolves have repeatedly used The same way a raven would go to a picnic bench where people go to a lot, or a garbage dump to find some food when it's hard to come by elsewhere. But why would a wolf kill site be predictable? Why would wolves kill their prey in the same place over and over again?
So this was really surprising for us. We thought at the beginning, well, a single kill is not predictable, which it is. I mean, we cannot predict when and where an animal is going to die. However, if we look on the distribution of kill sites of the wolves, we see they are not randomly distributed. They're clustered in certain areas. So if you look on the longer term, on a longer time frame, there is some predictability in the system. It's a similar thing like if you would visit as a raven picnic areas, there will not always be food in this place, but sometimes there will be. And it's not always exactly the same table, but it's in that area of the picnic area. Probably it's similar with wolves, just a bit on a larger scale. So they remember in certain areas, wolf kills are just more common, and then they just check it out. They fly there from large distances and see whether they find something.
What do wolf kill sites have in common? What's predictable about them?
At least in Yellowstone, we know this is from a form of papers on wolf behavior that kill sites, especially of elk, they're often in the flat open landscape. So in winter, typically snow covered. There's sometimes maybe roads next to it. So it's areas where wolves can be quite fast and pretty good in hunting. Maybe it's also some characters of the landscape that ravens could learn. It could be the presence of wolves. It could be previous feeding of wolf kills. We even discussed maybe they just find bones. So maybe in some areas there's a much higher abundance of bones. If the snow is not there in fall, they fly over the area and see, well, there's a lot of bones. It's worth coming back in a month, maybe.
I really like this description, this kind of comparison between what a wolf kill is like, where they're noisy, they're all running in a pack, they're howling, whereas a cougar kill, it's stalking, it's hiding your prey. And I could see how one might catch the attention of a nosy raven that's kind of hanging out near a kill zone. What does this say about scavenger strategies or how smart ravens are? Like now that you know that they're not following, but they're instead returning to kill sites that they remember or they notice.
So for me, this changes a bit how we think or how I think about scavengers. When we think about vultures, eagles, different corvid species, we always thought like, okay, a carcass is something very unpredictable. So how they would find it is maybe random search, is some social information transfer. It's just maybe sometimes just coincidence. They have to be opportunistic, fly a lot. And now with these new results, we suddenly can think about, well, there is actually something they can remember. So memory is much more important for scavengers than we previously thought.
Ravens, corvids are really notably intelligent. And we see this kind of behavior returning to sites in chimpanzees. So they're like, I'll go back to the fruit tree and see if it's fruiting again, or like a whale going back to a site with a lot of food that it remembers over time. Do you think we're going to see this in vultures or other scavengers?
So at this stage, we don't know whether any other species would follow this strategy, but I think it's quite possible. So especially species like vultures that depend on carcass even more than ravens do, I can totally see that they would also remember certain areas that are more profitable for finding carcasses than others. And I think it's super exciting if someone starts looking on that more.
One thing I wanted to touch on at the end here was you mentioned in the paper that the data is public. You can see where the ravens went, except for some of this predator location data. Why do you have to be a little bit more guarded with that?
For the predator location data, there is the issue that when wolves move out of Yellowstone, depending on the season, they might be hunted. So there is a certain quota. I don't know the latest regulation about that, but I know that a few years ago, 25 wolves were shot when leaving the park during one winter season. And this had drastic consequences on the social behavior of the wolves on the whole. I mean, some packs were completely destroyed basically by this. So this is why we cannot share this data because, well, would facilitate if some hunters really want to shoot wolves, that facilitates them to go to the right location to find them.
All right, Matthias, is there anything we didn't touch on from the paper that you think we should or that you want to revisit before we wrap up?
There's maybe something, I mean, it's not right in the paper. Maybe it touches it a little bit. Something I find quite interesting because the whole thing is about scavengers. And speaking about scavengers in general, they're kind of nature's cleanup crew. they remove carcasses from the landscape, and this can help to limit the spread of disease, because it's not always that animals die from predation, they also might die from diseases. And this is an important ecosystem service. And for this, they first need to be able to find the carcasses. And this is also exactly what we looked at in this study. So how are they able to find these food sources?
We definitely saw problems in India with this chain reaction where cows had drugs in their system, and then the vultures were eating those cows, and then the vultures were dying off, and then you started to lose that ecosystem service. And that's a real problem for people, for animals, for everybody. Better understanding scavengers is a really good idea. Thank you, Matthias. This has been really fun to talk about.
You're very welcome.
Matthias Loretto is an assistant professor in the Research Institute of Wildlife Ecology at the University of Veterinary Medicine, Vienna. You can find a link to the paper we discussed at science.org/podcast. And that concludes this edition of the Science Podcast. If you have any comments or suggestions, write to us at sciencepodcast@aaas.org. To find us on podcasting apps, search for Science Magazine or listen on our website, science.org/podcast. This show was edited by me, Sarah Crespi, and Kevin McLean. We have production help from Podigy. Our music is by Jeffrey Cook and Wen Koi Wen. On behalf of Science and its publisher, AAAS, thanks for joining us.